Welcome to Chapter 2, Biochemistry, Anatomy, and Physiology. For our first vodcast, you see from this title slide we're going to be looking at Chapter 2, some of the principles involved in biochemistry. First we'll start with classification. As you remember from chemistry, we have different groups of particles, atoms being the first, each with particular charges, protons, neutrons with no charge, and electrons with a negative charge. Most elements in biochemistry that make up organic tissue consist of very few, actually, inorganic elements. We use the acronym CHNOPS: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur make up almost 98% of the organic material, along with some other trace minerals and elements. If you take elements, again, bonded together, following this model, we can form molecules. Molecules are basically different atoms bonded together. Specifically in biochemistry, we do focus on carbon. Carbon's a very special molecule. Carbon, if you remember from chemistry, has the ability to form 4 covalent bonds, which are shared pairs of electrons. And we end up with this hydrogen-carbon bond called a hydrocarbon. This slide illustrates the 3 elements to atomic structure, the protons, neutrons, and electrons. You can see the 3 here. Hydrogen having 1 of each. Okay. Helium having 2 protons, 2 neutrons, and 2 electrons in the outer shell. Lithium in its outer valence shell having 1 electron, 2 in the inner shell, and an appropriate number of neutrons. With those in mind, we can then start to make even larger molecules of organic compounds. And we have 4 basic categories which those fill. Number 1, we have carbohydrates, which consist of carbon, hydrogen, and oxygen bonded in a 1 to 2 to 1 ratio. Glucose would be an example of that carbohydrate. You can see there a simple formula for glucose, C6H12O6, is simplified in a 1 to 2 to 1 ratio. Lipids, on the other hand, which consist of fats and oils, do not have that simple 1 to 2 to 1 ratio of carbons to hydrogens to oxygens. Other than that, they fall into 3 distinct categories. Number 1, triglycerides, which really provide as an energy source for our cells in our body, are composed of glycerol and 3 fatty acids, and I'll show you in just a minute an example of what these look like. With addition to phospholipids, we typically see that making up cell membranes, which is a phosphate group with 2 fatty acids attached. Lastly, we have a group called steroids, with cholesterol as a building block for those steroids. Steroids, if you remember, are very important molecules in our body that act as chemical messengers, typically affecting things like metabolism and protein synthesis. Speaking of which, proteins are our next category. Proteins are built of smaller molecules called amino acids. Now, there are approximately 20 different groups of amino acids that form human proteins. With these 20, though, we put them together in different orders, and we can affect a great number of human proteins. One characteristic of proteins is that they do contain both nitrogen and sulfur elements in their structure. The last group, which we don't spend a whole lot of time in our anatomy class, but you probably did in your biology class, are nucleic acids, composed of DNA and RNA. This group of macromolecules also contain nitrogen and phosphorus, and if you remember, are composed of items called nucleotides, which are containing sugar, phosphate, and a nitrogen base, typically found in a double helix structure. An example of a carbohydrate drawn 3 different ways. You can see the first diagram, what we call the ball and stick method, really gives you an idea about their chemical makeup, the empirical formula, but doesn't lead itself to help illustrate their structure. Diagram B and C help to do that, to give you some 3-dimensionality to these molecules, because that's important depending on how they bond. If you can see, for example, the hydrogen-carbon bonds here, and on the ends of these molecules, we call these functional groups. Now, it's the functional groups that really determine those characteristic properties for carbohydrates, how they react with other molecules and such. On the last diagram, you can see it's a very simplified diagram, removing all of the functional groups, and you have that figure that typically represents a molecule of glucose. One molecule of glucose, when combined with each other, can form a larger structure called a disaccharide. Okay, and comparatively with disaccharides linked together, we end up forming even larger macromolecules called polysaccharides. One of the next groups we mentioned are the fats and oils, and a triglyceride would be this example, which contains glycerin on the left-hand side here, along with 3 fatty acid chains on the top and middle and bottom. You can see that last fatty acid chain contains a special component, like a double bond. Okay, these double bonds do affect the general structure of those molecules. You can see here, this would be considered an unsaturated fat. The hydrogen has been stripped away right there, missing from that carbon, so it forces the carbon to form a double bond with its neighbor, essentially causing a shape change to that molecule. And we know when we change the shape of that molecule, we also change its characteristic properties. It's now becoming an unsaturated fat. If we were to do that a number of times with other hydrogens and remove them, we would end up with a polyunsaturated fat. You can see here a few other examples. We have our triglyceride on the left here. We have a phospholipid molecule, which would be simplified into this diagram here, composing a water-soluble head and a water-insoluble tail, which will characteristically form with other phospholipids, forming this double membrane that we call a phospholipid bilayer, which makes up most of our cell membranes. Lastly, remember the structure of cholesterol, forming many of our steroid molecules that are responsible for cellular communication. Proteins. Last group, or second to last group, are structure of macromolecules, or if you remember, are built from these smaller molecules called amino acids. Now, amino acids here you can see in this diagram are all linked in this very linear structure, which we refer to as primary structure, the first level of protein structure. Following that, we end up with a secondary structure. If you remember those functional groups, those side elements on those molecules can affect very weak forces and cause that molecule to coil, or in this case, pleat, like a folded piece of paper, causing that structure change we call a secondary structure. Next, we have this tertiary structure here, where these pleated or coiled molecules will start to, again, be attracted to certain areas on that particular protein. So again, shape is really important here. And then lastly, quaternary structure, where they may interact with other tertiary structure proteins, forming an even larger structure. Our last group of organic molecules are comprised of nucleic acids, both DNA and RNA. Very simply, DNA made comprised of nucleotides with a phosphate, a sugar, and a nitrogen-containing base. If you remember your base pair rule, adenine pairing with thymine and guanine pairing with cytosine forms a very complex code that provides the instructions. As far as proteins, a subgroup of proteins are enzymes. Now, enzymes function in the body to catalyze certain reactions. They really drive most of the metabolic reactions that take place, molecules changing into other molecules and releasing energy. Enzymes are formed from certain types of proteins. Now, these proteins have very specific shapes, three-dimensional in nature. And the specificity of each enzyme is dependent on that particular shape and structure of that protein. That shape can be altered by certain environmental factors. A change in temperature, a pH, a change in electrical conduction could all be factors in how that enzyme functions. There are also structures called, molecules called cofactors. Things like your vitamins, certain minerals, that assist how effective those enzymes are. And lastly, those enzymes, they allow reactions to take place at lower energy levels. We may have a certain metabolic pathway that would not take place at a particular level of energy, whether that be temperature, but using those enzymes allow those reactions to take place. Number 4, chemical properties. Most of these groups of large organic molecules have certain chemical properties that define how they participate in chemical reactions and with each other. Polarity, number 1, can result in the way certain molecules interact. For example, this water molecule, H2O, both hydrogens are covalently bonded to the oxygen. In some areas of that molecule have unpaired electrons, and those unpaired electrons produce a partially negative charge, where on the bottom of that molecule, the two covalently bonded hydrogens lacking those unpaired electrons produce a partial positive charge. And with enough of these molecules in place, we can form these weak bonds with other water molecules, really providing for something called surface tension in water and water cohesion. Water is a very cohesive molecule. Another chemical property that may affect certain organic molecules is pH. pH is defined as the negative log of the concentration of hydrogen ions. Now, it is a logarithmic change, meaning that a 1 unit change in pH level produces a tenfold change in the concentration of hydrogen ions. So with a few units change in pH, you can end up with a very highly concentrated acid or base. Water, being special, can be either acidic or basic, pure water being inherently neutral. Okay, and I have an example for you here. Using our graphing calculators, it's possible to find the pH of any given solution if you know the molarity. So in this problem, it says find the pH of a 0.025 molar HCl, hydrochloric acid solution. To find the answer, we simply enter into our graphing calculators 0.0025, and then we use the negative log to produce our answer, 2.60 pH. Okay, typically a 2.6 level of pH is a very acidic solution, which can affect certain categories of macromolecules, specifically enzymes. We've looked at that, how changes in pH can affect enzymes. We're going to come back to energy in just a moment. Here we see the pH scale from 0 to 14, with 0 and 14 being theoretical limits, and neutral being 7. A variety of substances that we are familiar with: apple juice, tomato juice, very acidic in nature. Distilled water, as we mentioned, being coming in at a 7.0. Notice human blood, 7.4. Human blood is highly regulated for pH, anywhere from 7.35 to 7.45. If we see a deviation from that interval for any reason, whether that becomes acidic, we call that property acidosis in the blood, or to the right, anything greater than 7.45, alkalosis. Usually will involve some kind of homeostatic change to bring that back into its set point. We mentioned energy just a minute ago. The cellular energy is driven by the chemical called ATP, adenosine triphosphate. Adenosine triphosphate is formed through a process called dehydration synthesis. Notice the precursors to ATP are ADP, adenosine diphosphate, and a high-energy phosphate group here. Using some energy derived from metabolism in the breakdown of our food, we end up with the formation of a molecule of ATP. In addition, we end up forming a molecule of water, hence the title dehydration synthesis, removing a molecule of water, producing a molecule of ATP. The antithesis to that would be hydrolysis. So again, ATP, in this case, fully formed ATP, combined with water, releases or cleaves that high-energy phosphate. If you see in the diagram below, the ATP has these 3 high-energy phosphates. Well, to drive cellular mechanisms, you typically need some level of energy. What happens is that high-energy phosphate is cleaved, forming ADP, and sometime later, using energy derived from metabolism and the breakdown of our food and cellular respiration, we can bind that high-energy phosphate back onto ADP, forming another molecule of ATP. We call this hydrolysis. Another factor that affects groups of organic molecules are the ability to form chemical bonds, otherwise called the combining capacity. Now, if you remember in your inorganic chemistry class, ionic bonds form due to the gain or loss of electrons in their outer shell, producing either positively charged or negatively charged molecules, or elements, excuse me. Number 2, covalent bonds we see more prevalent in biochemistry, deals with equally shared pairs of electrons in their outer shell. If you remember a previous slide, we mentioned carbon. Carbon has 4 electrons in its outer shell and has the ability to combine or share 4 additional electrons in its outer shell, forming 4 chemical bonds. In addition to covalent bonds, we also have polar covalent bonds. Depending on the three-dimensional structure of those molecules, the end of the molecules can become slightly charged, either with a positive or negative force. If you remember, we mentioned water being one of these elements that has that ability to have a polar covalent bond, and it provides for that cohesion. Molecules are attracted to each other with a very weak force. And lastly, hydrogen bonds, attraction between two polar molecules, for example, in proteins. We mentioned the protein folding, if you will, that allows proteins to fold into their secondary, tertiary, quaternary structure, really is due to these hydrogen bonds, these weak attraction between these functional groups that occur on the sides of the molecules. This ultimately determines the folding of those proteins. And here we see an example of a covalent bond between a hydrogen molecule and a hydrogen gas, one electron and one proton in the nucleus forms to produce this hydrogen gas. Hydrogen being number one element on the periodic table because of that arrangement of electrons and protons in the outer shell. Ionic bonds, however, as we see in this example with sodium, sodium tends to lose an electron in its outer shell, satisfying the octet rule, and joins the chlorine atom, producing a negatively and positively charged molecule here for sodium and for chlorine. And we have that weak attraction forming sodium chloride. Hydrogen bonds, an example here we see with this pleated sheet or coiled structure, the coiling or pleating of those proteins are really due to those hydrogen bonds, those functional groups on the sides of those molecules that form these weak attractions we see here, for example, in the coiled structure. Now, these weak attractions can be affected by things like changes in temperature and changes in pH and other environmental conditions that may disrupt that weak bonding attraction, and changing that secondary structure really affects the functioning of those proteins. As far as combining capacity goes, we typically see covalent bonds being the strongest, storing the most energy in those chemical bonds, all the way down to hydrogen bonds and polar bonds being very weak in nature and very easily disrupted by changes in the environment, such as heat and temperature. That's it for Chapter 2. Please go ahead and go back online the website and finish your vodcast questions, and we'll see you next week. Thanks.